exciting paper, at least. So we are going to talk about a paper which is published in the New England Journal of Medicine, published this year in January, called Lower or Higher Oxygen Targets for Acute Hypoxic Respiratory Failure. So I know I'm taking you to a very special setting. It's the intensive care unit. So acute care and so emergency medicine and intensive care are my research interests. And I know it's very specialized and for you maybe a bit unusual, but some aspects will be really new to you, but I will try my best to give you all the knowledge you need to understand everything. And I think it's worth struggling through some difficult topics like mechanical ventilation, which we'll talk about, because I think there's some really interesting aspects in the trial we can discuss here. But I know it's something you're not used to hear, so really feel free to interrupt me at any time if you're not understanding something. So I want to give you an impression of an ICU. So here's a picture from an ICU in Switzerland when the COVID pandemic started. Here you can see a physician and 2 ICU nurses with a patient who's about to be intubated, like the moment before it. And so he might be one of the patients in this trial, in the HUD ICU trial, because they included ICU patients with respiratory failure who are receiving high fractions of inspired oxygen. So, but what's the problem with this high inspired fraction of oxygen in patients with the mechanical ventilation? There are no clinical practice guidelines in the ICU, with a few exceptions, but there are no really broad guidelines. But there is clinical practice. But this clinical practice was not developed in ICU patients. Mechanical ventilation started in anesthesia. And there, the aim is to replace the lung for the time of an operation because the anesthesia causes a respiratory depression, which is not sufficient. And to replace the lung, the first big procedure is intubation. And there you give normally the maximum proportion of oxygen you can give before and after the extubation. So the oxygen flows through the lung into the smallest part. Here, the smallest part is called the alveoles. These are these things that looks a bit like little balloons. And these balloons are surrounded of blood vessel, here also in the picture shown, and there is where the gas exchange happens. But with the application of high proportion of oxygen, which is called hypoxia, there are some problems which might harm the patient. So due to the high affinity to oxygen of the blood vessel, the oxygen is drained out of these alveolus, and when it's filled with 100% oxygen, the alveolus is collapsing totally. And this is called resorption atelectasis. So you're collapsing part of the lungs, and then you can't use it for your gas exchange. This collapsing only happens when the alveolus is filled with 100% oxygen, but not happening if there's some other gas inside, like carbon dioxide. And now you have a totally collapsed alveolus, and you want to expand it as well. And for this, you need a high pressure. And this situation is often compared to the situation where you inflate a balloon. And in the beginning, you need a lot of pressure to start inflating the balloon. But this high pressure you need can cause structural damage to the lung. So one side note here, mechanical ventilation is always inflating oxygen with pressure during inspiration, but this is not how the body is doing it. The body is doing it passively. So anyway, we are applying pressure, and applying more pressure is more harmful. And when we are doing this due to high pressure, it's called lung injury. And lastly, with high proportions of oxygen, there are some reactive oxygen species which result in some oxidative stress. So what are the people doing in anesthesia? Because there it was developed, they just set the proportion of oxygen a bit lower, and so they don't have these problems. And but they don't have really the goal to go really in low values or normal with what you and me would have, because it's just a short amount of time. And if they don't have the complication, they don't really care what is the exact level. But this is a different setting. It's just some hours during the operation. But in the ICU where we are, the patient is ventilated for days. So we have to care for all these possible complications. So you got through this part. Now it's, I think, getting a bit easier for you. Any questions so far? Good. So let's go ahead. Now we are in the ICU setting, and there were some people who wanted to test this setting. So we are always talking about how it's done in anesthesia, so some higher levels comparing lower levels. And lower levels are represent a bit more the normal range, so persons without mechanical ventilation. And there were some 3 studies. First, the oxygen ICU study. There, they could show a mortality benefit in lower oxygenation targets. Then it was the ICU ROX. There was no difference in mortality. And there was the LOC2, and there was a higher mortality in the lower oxygenation target. And in the lower oxygenation target, we had more adverse events concerning mesenteric ischemia. This is when the intestine doesn't get enough oxygen and is dying. And this was the reason why the study was early terminated. And now we have the perfect situation for another trial, because we had 3 trials with 3 different results. And that's the reason we needed the HUD ICU trial we are discussing today. So now it's your part. I hope some of the students can tell me anything about the design of the study. So feel free to talk about setting, allocation, concealment, masking, trial type, anything what comes to your mind. Oh, sorry, I'm giving you a peek. So it's easier now if you're a quick reader, but anyone, please speak up. I hate to call out people, so just some volunteers, please.
Setting, obviously it was ICUs, so quite specific, patient type, as opposed to, some trials that are set in more general, broader settings in the hospital. So ICU is very intensive, and I think it was international. So that helps. Trying to think, masking. I don't remember where they said blinding, but sort of, okay, so single assignments can't be blinded, but they could have blinded analyses, and I believe they did. So that's sort of something that, even if people can't be blinded to the intervention, it's always good to blind the people doing the analysis, and you can pretty much always do that unless they're like really involved in designing and assigning the people to the groups.
Yeah, thank you. Great points. So what I had also in mind, I will just give you my thoughts. So like I said, RIAS, it was international, multi-center randomized controlled trial. For the allocation concealment, they used computer-generated allocation concealment with permuted block randomization. Like you said, the outcome assessors were masked, the treating team was not masked, and the trial type is a superiority trial aiming for an absolute risk difference of 5% at 90 days. And I'm going to talk about all the bolded parts of the design and discuss some points here. So what is the purpose of randomization? Rias saved one question, so somebody else has to take this question. Anything what comes to your mind?
So if none of the students can respond to this question, I think we're going to have to revoke your status and send you back for a repeat year. This should be like the most basic. You should all be leaping to answer this question.
Doug, I gave a talk on bias and randomization a few weeks ago in the Biostatistics Slam. I invited everybody to come to it. I don't know who came. I couldn't tell. But anyone who came to it would know the answer to that question.
So now the pressure is really high. Maybe I can take it off, because now if you're saying something wrong, it's really embarrassing. So I'm taking this over. So but be prepared. There are some other questions. You're not done for today. So I was thinking about preventing selection bias, to have comparable groups between treatment and intervention, control and treatment, and for your statistical methods there you can use random sampling assumptions. So let's check for these purposes. Let's talk about selection bias and random sampling. Here I gave you figure 1, so the participant flow, and highlighted a thing I wanted to show you. So we have a 4,000 patients who were screened, and a third of them were excluded. So there's maybe a discussion point if we really have random sampling, but yeah. But one other thing I really want to show you here is one point here has no provision of consent. And this is an interesting feature. I know it's had nothing to do with randomization or sampling, but it's mentioned here on the slide. So I'm gonna talk about this, and it's very specific to the setting of acute care. And when you think just 295 had no informed consent, which is a very small number considering what are the patients investigated here. So they're all sedated, they're unconscious, so they can't provide consent. And since respiratory failure is often very acute, you can't plan for it and discuss it with the patient beforehand. And I think they had here a very interesting solution, which I wanted to highlight. Or does anybody from the audience want to share with us? So, because the patient was unconscious. Go on.
Yeah, so they use if the surrogate is available, they use a surrogate. If the surrogate is not available, they use, I believe that's doctor independent of this trial. And after the consent, and if the patient became available or the surrogate became available again, then they have the choice to withdraw the consent.
Exactly, perfect. That's the answer I was looking for. And I think in the setting, it's a very good solution. And so they could collect a lot of data. When the consent was withdrawn, just one note, they asked the patient or the surrogate if they can continue registration or if they can use the data so far on, which I think gave them also a bit more data. So next point of randomization was comparable groups. I don't want to go into any detail. This was a long table 1 comparing lower oxygenation group and higher oxygenation group, some baseline demographics, something about illnesses. Here's something about the ventilation parameter, and some very ICU specific things about how ill the patient was. But I think they did, so randomization did a quite decent job. I think the distribution is quite comparable. So to the next point.
Can I make just a point or a question that, because I think if you can go back a couple slides, it's just because it popped into my head, sort of the idea of the random sampling versus random allocation, right? Randomization is it's sort of, it's preventing in that selection bias in getting at who is assigned to A or B, who takes A or B kind of thing. But I think everybody generally agrees trials are still quite a selective sample, and there's still a selection bias in the people that they're taking. So it doesn't necessarily prevent, I think selection bias in the sampling is actually pretty much largely there for trials. That is sort of one of the main sources of that. It was just sort of on the previous slide when you were talking about, you know, having these people, or maybe one, yeah, sort of people being excluded if randomization was doing a good job of getting random sampling. But I don't know that, you know, it's still going to be a selected group.
Exactly, you're right. That's why I phrased it so strongly. If it's completely prevented, I think we can't completely prevent it.
Yeah, but just, you know, to kind of echo Riaz's point, I think the random sampling you're referring to has more to do with generalizability than the validity of the trial.
Mhm.
Yeah,
Thank you.
can I chime in on that? I'm more under the impression that that would be referring to the sampling bias rather than the selection bias. Yeah, because we tend to associate selection bias as having uncomparable groups, like in our sample in the trials, instead of like how like similar the sample in our trial is with the target population that we are aiming to like make any conclusions about the validity of the treatments. Yeah, it's just a impression.
Yeah, and I think with so comparable groups wise, I think it is quite comparable.
Yeah, I just have one question. So I wasn't reading too much in details about their randomization scheme. Did it use blocking? Because I see a lot of pneumonia cases, and that trial happened right around the time of 2020, I think. Was it?
It was also covering the COVID pandemic, yes.
Yeah, so if they failed to, if they didn't like take secular trends into consideration, that the COVID patients might be a problem if, you know, they randomize some patients to one group more before the pandemic and maybe more into another group after the pandemic. So blocking would be some features that is really like safe, kind of protective against the secular trends in this case.
They mentioned the blocking and the stratification, but that's,
yeah, okay, good.
And I think they wanted to have this broad overview of all ICU patients, so not to pick one specific subgroup or to focus on it. So this is really the problem in the ICU, this heterogeneous group they are treating, and you're treating a real different patients with the same treatment. This is one of the problems also here, but in general, not here trial-wise, but setting-wise. But you're right. So thank you very much for all this input. I'm going to the point about masking. And I want to explain you why the treating team couldn't be masked. I have to explain a bit about mechanical ventilation there and how they achieved their difference between groups. So stay with me, but we're coming to masking. So if you're looking at the protocol registered, for example, at clinical.gov trial, and there you have to name a treatment or a drug for the intervention and the control, actually both are oxygen for intervention and for control. But their target was a difference, but there wasn't a difference in the amount of oxygen. Their target was in the blood. So just to look at this scheme, it's very basic, but here we have fresh gas. So here the oxygen is coming out, going into the patient, into the lung, again in the alveolus I was talking about, and then it's going into the blood. And there you can measure the partial pressure of oxygen in the blood. And that's actually what they are talking about. So the partial pressure of the blood is dependent on how much oxygen flows into the lung. So it's the fraction of oxygen going into the lung, but additionally, it's depending on the proportion of oxygen going out of the lung. And we can regulate the outflow, and this can be done when you add some pressure when breathing out, and breathing out is the expiration, and this pressure is called PEEP. People who are working with COPD patients know this, but I don't want to go into detail. I think this basic concept is fine here, telling that you can vary this partial pressure of the oxygen by the fraction inspired and also by the PEEP. And but you can't set a value for the oxygen and a value for the PEEP and then just be sure that it will get the partial pressure you want. So it's not predictable and you need to adjust it. And actually I can show this, that it's not depending only on the fraction of oxygen in the trial data. So we are having here some results. Here is the main results of the groups. We are going to talk about this in a bit later as well, but here we have the partial pressure of the oxygen in the blood, and we have the days of the ICU. We have the two groups, the higher oxygen target in red and the lower in blue. And they are giving us the data of the inspired fraction of oxygen, so how much oxygen is going inside the patient. And here you see that it's quite an overlap. So you can't say with a fraction of 50%, I will achieve the lower or the higher target. And that's the reason it's not predictable. They had to adjust it. And that's the reason the treating team couldn't be masked. And now, but we can see if they did a good job in adhering to this, to doing their job. And here I just highlighted the aiming targets. For the intervention it was 60, for the control it was 90. So for the lower oxygenation targets is a bit above what they aimed for, but I think still they did a decent job, because you can see a difference between the two groups. It's getting a bit washed out in the end, but I think it's also depending, because not a lot of patients are staying at mechanical ventilation shots for a long time. So now I wanted to use this study to just talk about trial types, because I wanted to push myself and think about other types of trials. Here we have the superiority trial, but are there any alternatives for this? Now I need some assistance from the audience. Just in general.
I mean, I guess with regard to hypothesis or with regard to actual design kind of thing, or both, all of the above. Yeah, hypothesis, you know, if there had been, I was kind of wondering why, you know, at first before reading that there were some potential harms associated with higher target. I wondered why it mattered really. So they could have done a non-inferiority or something as well, or equivalence, but that would be more like if there were some, generally I think if there were some reason why they wouldn't want to just do a superiority, why they didn't think that mattered. Trying to think what other kinds of trial designs. It's kind of not really good for a crossover, I don't think, because you're sort of got something that is going to affect how the patients are, sort of their baseline over time, right? And sort of it's an intensive care unit with sort of an acute thing. So putting somebody onto something and then switching them around, I don't know that you wouldn't have some residual effect from before. So parallel may be the best option, as they've done.
Great, it's like I told you what I was expecting to hear, so perfect answer. Yeah, just the non-inferiority and equivalence. And just like you said, you have to adjust your objective and your hypothesis to this. And I thought maybe we can do this just as an exercise, a mind exercise with this trial. So we have this superiority design, and there the objective was low oxygen targets for acute respiratory failure are better than higher oxygen targets. So we have a lower mortality within the low oxygen target. And we're testing the null hypothesis that the low oxygen and high oxygen treatments do not differ. And the alternative is that there is a difference between low and high. And if we are going into the non-inferior design, there we would say that the low oxygen targets in respiratory failure have no higher mortality than the high ones. And we're testing here a null, or one of the nulls we could test is that low oxygen targets have higher mortality than high oxygen treatments. And it's a one-sided test here. But you said, like you said, it's just what do you want to ask, and is it appropriate to think about this way? And while reading for preparation, I saw these non-inferiority assumptions. So you have to have a proper control arm. The data should be available. You should have some constancy over time and among patients, and you have to perform an assay sensitivity to demonstrate a true difference. And I was thinking if they could really done this way. So should they have used a non-inferiority trial? And what I was thinking, like I explained to you in the beginning, it's not really that there is a control arm which is proven to be better. There are no data available from the control. So I think at least two of these, or the first two assumptions, would have been violated because they choose the control arm using the clinical practice and not data. So maybe it would be a bit a stretch of these assumptions. But I want still to talk about the non-inferiority trials, because I think it's interesting here in the setting. And when you're talking about the in non-inferiority trials, there you always have to think about the interpretation. And I really like this figure from the Fundamental of Clinical Trial textbook. And I think maybe it's worth it to just discuss it once again. So here you have the relative risk. Sorry, my pointer doesn't work that well. From 1.0, so no difference. And now we are looking at the point estimate and the confidence interval. And this delta is the so-called non-inferiority margin, which is determined by the researchers. So we are saying, what is the delta? We are thinking the new treatment won't be different, so we call it non-inferior. And here are the two options of a non-inferiority trial. So you can have the point estimate above 1.0 or below 1.0. The important thing is that they are below this delta and that they are containing 1.0. If you have the opposite, so you are containing 1.0, but it's crossing the delta, then it's inconclusive. And if it's not containing the null value of 1.0 and containing delta, it's inferior. In a non-inferiority trial, we could show superiority, and that's this case. So the point estimate is below 1.0 and the confidence interval does not contain 1.0. And another thing I was reading about was some sample size consideration you have to take here. I did it by hand without any program. So if anybody sees a mistake, just shout out. But I wanted to just use it as an exercise. So these are actually the values of the trial. We have this 90% power, alpha 0.005, and detectable difference at 5%, 90 days mortality. And there we have the N of 2,928, which also they used. And here you're having the control versus treatment, which is different in the non-inferiority design. So don't be confused. It's if we would have always the same denominator and determinator, it should have been switched. And I just made the calculation. If we say that 5% are fine within the difference of mortality between the groups, the sample size is a bit lower. I think it's because of the one-sided test. But if you say, okay, the researcher think that 5% is superior, so maybe we should just have a lower inferiority margin, then you see that the sample size is just blowing really high. So it's going really up. So enough mind thinking hypothesis, just go back to the trial, but I saw that Janet Holbrook unmuted herself, so maybe there was a fault on this.
First I was going to say another alternative for trial design would be a group randomized trial where you just randomize by ICU. And then with your slide that you just went over, I was, no, the one you were on, just wondering about the alpha and whether for a non-inferiority trial, we really should have a alpha of 0.025 to have the same kind of certainty as we would have with a two-sided test. And that was really a question and not.
So to be honest, I don't have any experience, so maybe there some faculty could chime in and discuss this.
I was wondering if Jay had an opinion.
Yeah, good question, Janet. But I think in case of a non-inferiority, let's see, for superiority design, okay, Alexandra, for the superiority design, you have that as two-sided, right?
Yes.
Okay, so in the non-inferiority, it is a one side, both of them should be one-sided. So, but yeah, I think you could have alpha 0.025, isn't that right? Yeah, because they're one-sided tests, and to be equivalent to the superiority design, I think Janet is right. But you see, so what would that do to the sample size? One-sided 0.025 would require a even larger sample size, right?
Yeah.
Yeah. Okay, we can look at that later, but I believe I always get mixed up with non-inferiority designs. I detest them because everything is backwards. But
Yeah.
Janet is correct here.
So that's reassuring because I spent a lot of time thinking through, and I was thinking maybe I'm doing some mistakes. So it's reassuring that it's not only me having to struggle through this, but everyone is a bit having to think about it twice or even more times.
Yeah. So, I mean, I agree with Janet, but at the end of the day, people do what they do, and seems to me I see a lot of I do see non-inferiorities with an alpha of 0.05, but I think Janet's right. To have the same certainty of the on the data, it would be an 0.025, and that would balloon the sample size. So, and that would, and I think Alexandra's correct. The reason why her sample size is pretty close or slightly smaller with the non-inferiority design is simply because it's a one-sided test on the same alpha when it should be a one-sided when it would be much larger if it was a one-sided test on the alpha of 0.025. So anyway, probably should move on.
But thank you. I learned a lot from this discussion, so thank you very much. And to go to the results, here you have the primary outcome of mortality with the proportion being very similar, about 42%, and the risk ratio adjusted for the stratification variables or adjusted for stratification variables and baseline variables, so in the adjusted odds ratios, are a bit above or 1, but containing all the null value of 1. So here we have no statistical significant difference from null. When you look at the secondary outcomes, also not really a big group difference, except maybe when you look at the subcategories of the adverse events, there you might have the impression that it's not really the same, but the event numbers are very small. So maybe we should push this conclusion too much about the adverse events. But talking about conclusions, there were some other people doing some conclusions about this trial, and I just wanted to show you some of the media response after the hot ICU trial. So here, this is the bottom line. It's a British ICU blog, and they are always talking about some different studies and evaluating the results for clinicians to briefly read through. And there the author is saying, while this trial does not change my practice, it's reassuring to see that the targeting a PO2 of 60 millimeter Hg for patients with hypoxemic respiratory failure does not result in harm. And I will continue to use the minimal FiO2 possible to maintain my patient's SpO2 above 90 whilst awaiting further trials. And let's look at another blog, that's Rebel EM. It's about emergency medicine and acute care. And their clinical take home message of the study is, this study adds very little information into clinical practice and does not provide a definite answer for specific oxygenation parameters. Current evidence show that we should embrace lower PO2 and SiO2 to continue to wean FiO2 as quickly as safely as possible while ensuring not to expose patients with suboptimal oxygen saturation. And we need larger trials. And some Twitter comments about hot ICUs. One is saying hot ICU is not showing worse mortality. So the one here, outcomes in those with, so that's just the other unit above 8 is here 60, and this is just the other unit. For those in acute hypoxic respiratory failure, another saying, take-home message, current pandemic NIH practice limiting oxygen use appears safe, results consistent with ICU ROCs, extra data needed on some subgroups. And one, the last one, so provides high quality data for patients with respiratory failure, and the 2 oxygenation targets lead to similar outcomes. So after reading this or listening to this, would you agree? And it's the last question, last opportunity.
I think it's, personally, I think it's an interesting statement on interpretation, right? I mean, technically, I don't think there was anything really wrong that jumped out about the different ways of, saying one is no worse, one is no better, but just that people choose one as opposed to saying, that people, choose to highlight one specific one in their statement about it, right? The takeaway is that 92% is good, or the takeaway is 60% is good, and it's not going to change my practice, as opposed to sort of highlighting the overall, that you can use either, just sort of show people's inherent internal bias. People will take away from it what they want to.
So I have a kind of a question or comment. If we put this trial in the context of the support trial, which attempted to answer a somewhat similar question in premature infants, basically whether high or low oxygen targets were helpful or not. There are different counterbalancing risks in infants. Their trial did find a difference between the 2 oxygen groups, and one of the consequences of that trial, which became quite publicized, is that the consent form in the infant trial did not talk about the potential risks in either of the oxygen groups because they were both felt to be clinically appropriate. The same is true here, that the patients were both being targeted within a range that clinicians felt was appropriate. I wonder if anyone knows, I'm guessing we don't know, whether the consent form talked about the potential for a higher mortality in one of the groups versus the other, because that was a significant criticism of the support trial. Any, Alexandra or anyone else, was there any discussion of that?
So I was one of, so actually in my last ICU where I worked as an MD, it was a side of the hot ICU trial. And if I'm remembering correctly, it was not discussed on the informed consent.
Interesting.
But I'm not 100% sure because there was a different protocol for the independent physician and for the surrogate. So I'm not 100% sure.
Interesting. You know, if this trial had found an important difference between the two groups, you could have made the same argument that was made against the support trial, which is, of course, you could have anticipated there would be a difference, and one of the groups therefore did worse than the other. How is it not okay to warn patients that one of the groups might actually do worse than the other? That's the whole controversy about the support trial and the ethics of it. That was done here in the US, and there was actually a regulatory action against the investigators. Maybe in Europe, they have less concern about getting sued or regulated also.
Yeah, it's always obviously a question of what you put into your consent form. It's stickier when your hypothesis is a higher mortality in one group than the other, and all the preliminary data suggest it. Then you have a real, you must have a detailed discussion of that. It probably would be appropriate to have some discussion of it, but remember the 3 trials that led to this show higher in one, no difference in higher in the other. And so you have a hypothesis, but you have preliminary data to support any hypothesis. You should, I believe, in a consent form where mortality is an outcome, discuss what the outcome is and why it's important. But I wouldn't even know what to say about the hypothesis here, other than the fact that mortality may be different between the groups.
Right. Well, so in the infant trial, they didn't even say that. The only risk that they talked about was the risk of irritation from the tape that's used to hold the pulse oximeter in place. Yeah. And that's a common kind of way of approaching the description of risk. Mm-hmm. But there are people, and I think I'm among them, that argue that if you're using mortality as an outcome, you have to at least wonder whether one of the groups will have a higher mortality rate than the other. And I don't know how you don't describe that, or at least, mention it as a potential risk. But anyway, I just think in these critical care studies, it's very difficult to, you can't do consent the same way you would in other situations either. And there's a time pressure that's not always present in other kinds of studies too.
Yeah. Well, what's even worse would be a cardiac arrest study, because it's almost impossible to deal with consent issues in a cardiac arrest study.
Sure. Same thing for field trials of, emergency treatments.
Yep. So
I guess would one way be, because, you have to have that hypothesis that one will be different as far as, death kind of thing. If that's if it's like your primary outcome. But if both are used in practice and really people, it's a toss up whether you get a doctor who goes one way or the other way. Is that sort of where you would focus your consent? Then you'd say, one might be different and that's why we're doing this, but right now they're both used.
If you're asking me, I would argue that you should say both approaches are commonly used by clinicians. We don't know whether one is riskier than the other, but there's a chance that one of them could be. Yeah. But people are, clinicians and researchers are hesitant to say that because they're concerned it will scare people away from the research. I would also argue that in a study just like this one, when you're targeting an oxygen level, you are to a certain degree tying the hands of the treating clinicians They're not going to play with all the knobs and levers that they have available on their ventilator to the same extent that they wouldn't outside of the study, because they're trying to hit some sort of oxygenation target. And there could be unanticipated consequences of that also. And I think that was also seen in the infant trial that I'm mentioning. So, but I agree, those are risks to the trial. If you over describe the potential risks to participants, you won't have a trial, and then you can't answer a really important question.
Yeah, I think mortality is the primary outcome. Consent form needs to mention mortality.
Yeah.
Where you get into stickier issues, and we probably don't want to spend a lot of time on this, is where it's not the primary outcome, but it's an outcome because it might occur, such as an AIDS trial, right? But not so much now with the efficacy of modern antiretroviral therapy, but at the height of the AIDS epidemic, any treatment for an ancillary disease or an infectious disease could lead to a mortality difference.
Right.
Yet the outcome is not mortality. A mortality difference would be an unanticipated difference. And so you might not put mortality in your first consent form, but after in your second trial onward, you would if you had a mortality difference in your first one.
Sure.
So, and it's always the disclosure of risks have to be based upon a sort of notion of reasonableness. What would a reasonable person want to know in order to decide to sign up for the trial?
Well, I just, in case not everyone's familiar, in the infant trial, there was a regulatory action by the Office of Human Research Protection, which criticized the IRB and the principal investigators for not disclosing that potential risk. And it became a very big deal. The clinical research community violently rejected the government's stance. There were editorials in the New England Journal, including by the editor-in-chief, which had published the original article. And I don't think there ever has been a good resolution of what the clinical research community thinks ought to be done in trials like this one. I don't think that we know. If this trial was done in the United States, I don't know whether or not OHRP would complain. Now, they didn't see a mortality difference, which they did in the infant trial. That's an important distinction.
Yeah.
They didn't know that beforehand.
Right. Well, so the other thing is, which I would say about this trial, I think for the type of trial it is, they did a pretty good job. And the results are what they are. There's no difference. They detected no difference. The issue then is, what are you going to do in clinical practice? Is one harder to do than the other? Is one more costly than the other? I mean, there are no, as far as I can tell, not being an ICU physician, it becomes a regional preference, a pick'em. And I don't know of any other reason to choose between the two approaches. So, and, Alexandra was in an ICU prior to this, so maybe she has some insight as to what might be a choice. But, and when you read the article, you didn't come away with what do you think we ought to do? We came away with, well, this is that. And I guess the other thing everybody should remember is, in normal people, if I remember back from physiology, which was a long time ago, the PaO2 is between 70 and 100. So 70 and 90 are within the normal range. And so we're targeting the high end of normal and the low end of normal.
To follow on that, I wrote down as Alexandra was doing her presentation that when she showed that the upper end actually met its target, but the lower end didn't really seem to come as close to meeting its target. In other words, the two groups weren't quite as far apart as the targets would have been. And then they got a nil result. Is it surprising that perhaps they didn't have enough separation between the groups? It looks separated, but it wasn't perhaps physiologically separate enough. And, maybe it was hard for clinicians to hit that lower target because they saw other things that made them nervous. I'm not going to turn down the PEEP any further because I'm worried about this patient or something like that.
Yeah, I was going to say that one of the conclusions of this trial might be that it isn't practical to make the separation that was the original protocol intent, and that might be a better, more comprehensive conclusion than just that there is no difference between the groups.
Right. That might be right.
Yeah, and although they didn't hit their target, and that is true, they came pretty close, and they did have clear-cut separation. So I think the conclusion might be that within the physiologic range, doesn't matter whether you target the high end or the low end.
Yeah, okay.
Yeah, just the range was it's the normal range. You're right, and what you talked, it's about where you are. So I was first taught at one side where you're targeting the lower end, and then I switched to another where they preferred the higher end. And me as a resident, I was wondering what to do because I didn't know what to do. One was saying this and the other saying this. So it's exactly what is happening. So if there are any other urgent thoughts, I will thank you all for your attention and for the discussion. And I hope you enjoyed the paper and the journal club. Thank you very much.
Thank you. Great.
Thank you. Great
Thanks a
job.
lot.
Alexandra, can you stay on for a minute?
Yeah.
Well,